Good day, everyone. I am Eva May V. Alahas, and I am here to explain the cell cycle, focusing on the stages of mitosis and meiosis and their differences. Mitosis is a type of cell division that occurs in somatic or body cells. This type of cell division produces two identical daughter cells which are ideal for growth and repair. It is composed of four stages plus cytokinesis. The following are the stages of mitosis. Prophase. In this stage, the chromatin starts to condense, forming the individual duplicated chromosome. The nuclear envelope or membrane starts to disappear or disintegrate as the mitotic spindle of microtubules develops from the centrosome. Metaphase. The microtubules extending from opposite centrosome attach to the kinetochore of each sister until all duplicated chromosomes align in the middle cell or the metaphase plate. Anaphase. The microtubule attached to the kinetochore of each sister chromatid contracts until the connection of the centromeres of the sister chromatids breaks. Thus, each sister chromatids are separated. Telophase. As each member of the sister chromatids reaches the opposite poles, the spindle fiber disappears and the chromosome uncoiled and entangled into chromatin. Then, the nuclear envelope starts to reform. Cytokinesis. The cytoplasm divides halfway along the length of the elongated mother cell. In plants, cytokinesis is initiated through cell plate formation or the fusion of Golgi-derived vesicles which contain new cell walls and cell membrane substances. In animal cells, cytokinesis is mediated through cleavage or contractile ring formation which is governed by both astral and central spindle microtubules. As the final product of mitosis, two identical daughter cells are formed. These cells are identical since they received one of each sister chromatids from the parental or mother cell. Meiosis. Meiosis is another type of cell division which occurs only on specialized cells called germ cells or non-somatic cell. This meiotic cell division produces four unique haploid cells. This type of cell division involves one DNA duplication, interphase, and two karyokinesis nuclear division and cytokinesis cytoplasmic division. The first phase of meiosis involves the separation of the homologous chromosome, resulting in the reduction of chromosome number per daughter cell. Thus, this phase is also known as reduction-division meiosis. First phase of meiosis consists of the following stages. Prophase 1. This phase has 5 subphases or stages, which includes the following. Leptotin or leptonema. The chromosomes thicken and become visible, but the chromatids remain invisible. The centromeres begin to move towards the opposite poles. Zygotin or zygonema. Homologous chromosomes enter synapses, the fusion of homologous chromosomes forming the synaptonemal complex. Pachytin or pachinema. The synapsis is complete throughout each pair of homologous chromosomes. Crossing over genetic exchange between non-sister chromatids of a homologous pair occurs. Diplotin or diplonema. The synaptonemal complex dissolves. The tetrad of 4 chromatids is visible. Crossing over points appears as chiasmata, holding of non-sister chromatids together. A meiotic arrest may occur at this time in many species. And diakinesis. The chromatids thicken and shorten. At the end of prophase 1, the nuclear envelope breaks down and the spindle fibers begin to form. Metaphase 1. Tetrads line up along the metaphase plate. The chromosome of a homologous pair attaches to fibers from the opposite poles. Sister chromatids attach to fibers from the same pole. Anaphase 1. The sister centromeres remain connected to each other and chiasmata dissolve. The homologous chromosomes move to the opposite poles. Telophase 1. Each complement of the homologous pair reaches the opposite poles. The nuclear envelope reforms. Cytokinesis separates the daughter cells. The end product of first phase of meiosis are two daughter cells with half the number of chromosomes, concerning the original cell, each consisting of sister chromatids. The second phase of meiosis contains the separation of the sister chromosome, which results in an equal number of chromosomes per daughter cells. This phase is known as equatorial division meiosis. This phase consists of stages similar to mitosis, except that there are two cells simultaneously engaged in the same stage. Prophase 2. The chromosomes condense as the centrioles move towards the opposite poles. The nuclear envelope breaks down at the end of prophase 2. Metaphase 2. Each chromosome aligns at the metaphase plate. The centromeres of each sister chromatids attach to the spindle fiber from the opposite poles. Anaphase 2. The sister centromeres detach from each other, allowing sister chromatids to move to opposite poles. Telophase 2. Each sister chromatid begins to uncoil. The nuclear envelopes reform. After completing the meiosis, there are four daughter cells produced, each having half of the chromosome number with respect to the original cells. These cells are called haploid cells. Mitosis produces daughter cells which are similar to the original or mother cells. This process is very useful in damage repair mechanisms, growth and development of other tissues, organs, and different systems in a living organism. Conversely, meiosis produces four haploid cells, half of the chromosome number with respect to the original cell. Meiosis is involved in producing gametes. Each gamete is unique from each other, even though they originated from a single cell. Gametic life cycle. This is common in the animal and some protista. The term gametic refers to the result of meiosis, which is gametes. The organism in this life cycle is diploid. During gametic life cycle, growth and development of diploid individuals, production of the reproductive cell of diploid individuals, formation of gametes, haploid cell, egg or female gametes, and sperm or male gametes through meiosis, and the fertilization of gametes, egg plus sperm, forming diploid individuals.